So in this last video, we're going to finish up reading our paper. We just finished working our way through all of the data in the paper. That's what you're looking at when you look at the figures and the results. And then again, we referred back to the specific sections of the materials and methods that we needed to in order to understand the data that we were seeing presented. The very last written section of the paper is what's known as the discussion. So if we go back to our little chart here, our little list of the sections of the paper, there are several things that the authors want to do in their discussion. So one thing they're going to do is a quick summary of the paper, talk about what they did, talk about what their major findings were. They're also going to talk about how their results compare to things that they talked about in the introduction. Do their results support what's already known? Does it contradict and suggest a new paradigm? So they're going to take what they did and now discuss it in the context of things they already brought up in the introduction. And combined with that, what they're really trying to do is put their research into the broader context of that particular field. So what are some of the implications for this, and what does this tell us, or what does this add to the body of knowledge that we already have? One thing that a really good discussion should do is point out any limitations on their study or any potential contradictions that they have, things that they couldn't quite explain. Maybe perhaps some of their results contradicted each other. They would need to address that. And then hopefully at the very end, they talk something about the practical implications. So how do we take this study, and this is a study in the case that we're looking at today, looking at bacterial concentrations on gym equipment. So they'll first discuss it in the context of what we know about bacterial contamination on various types of equipment. And then when they get it to the practical implications part, okay, think about what does this actually mean in terms of practice and human health. So sort of really broadening out the scope. So let's go back to the paper and kind of point out where we would find this information. Again, typically there's going to be a particular order to this. You're always going to start your discussion with a quick summary. Then you'll have these three areas, how it compares to the introductory information, so what's already known, the broader context within their field, and then the limitations and contradictions. That order might be slightly varied. And then it typically always ends with some sort of practical implication. All right, so let's go back to the paper. And if we just want to start highlighting, and let me just quickly change the color here. So all of this is talking about their specific study. And what they found, right? This study examined the ability, and our findings suggest. And then they're going to get into some of the other things that we talked about. So notice even in this sentence here, they're starting to cite additional research. And then all in here, what they're doing is they are discussing, so I'm just going to change this to a different color, a related color, discussing their results. So notice here they're referencing table 1, and we'll keep going on to the next page. They're discussing their results in the context of what's already known. So here it says the most common bacterial genus was found to be Staphylococcus. That's one of their results. And then they say these findings support previous studies. So they're talking about ways in which their results agree with what is already known. If we go back to our list of things that we should find in the discussion, remember a good discussion should identify any limitations or contradictions that they find in that paper. And if we go back here, I'll just highlight down here. One thing they note in their discussion section is that they were unable to detect any methicillin-resistant Staph aureus, which was a type of bacteria they were particularly interested in. And so what they note is that they had a limited sample size. So what they're doing there is they're doing the correct thing, and they're pointing out limitations to their paper. And then again, going back one more time, after you identify limitations and contradictions, you've put it into a broader context, the last thing they're going to do is try and identify a practical implication for their paper. So why should you care about this result? What does it mean for you? And if we go back here, we'll scroll up to the top, and right at the end of their discussion, they end with this sentence: implementation of infection control strategies such as the use of copper alloys in combination with disinfecting procedures should be considered to augment cleaning protocols and mitigate the spread of community-acquired infections, or CAIs. So what they're saying is gyms are already using disinfectants to clean their gym equipment and other cleaning protocols, but what they would recommend is that you implement the use of copper alloy, so copper-based gym equipment, to reduce the bacterial burden, the number of bacteria on your gym implements, and that could potentially reduce the number of community-acquired infections, fewer bacteria, fewer illnesses. And then right after that, there is an acknowledgement section. This is optional. Sometimes you'll see this in a paper, sometimes not. But what every single paper would have is their list of references. And so these are all of the different peer-reviewed sources that they used in their introduction and their discussion. So if we want to kind of go back here and finish this up, remember the discussion will also be very heavily cited. They're going to be adding lots and lots and lots in terms of references that they're combining with their results. So your introduction and your discussion will be the most heavily cited sections of the paper. The results should have typically 0 citations because you're presenting original data that you actually generated. Sometimes you'll have a citation in your materials and methods if you're citing a paper that provided the method that you used, but typically that also is completely written out and doesn't have nearly the amount of referencing that the introduction and discussion does. So just to leave you with a couple final tips. So remember, specific sections of the paper can give you specific pieces of information. If you want to know how to repeat the study, you go to the materials and methods. If you want to get into their data, you're looking at results and then the figures and captions that go along with that. If you want to know what are the implications for their work, you go to the discussion. Also remember that when you're in specific sections, there's typically specific places you can go for the information that you want. Always think back to this introduction where they say typically follow the same format of background information, justifying their study, what they did, and again, typically what the major finding was. And then also the main thing you should take from this is not to read your paper straight through, but instead to read it figure by figure, where you're sort of mentally putting together the specific materials and methods and results sections that go with a particular figure or table. This concludes all of the videos for how to read a scientific paper. At this point, you should be ready to get out there and start reading and complete your assignment for this week.